Microscopes, telescopes, and other technologies. Video 1 for cell structure and function unit. Both of the microscopes and the telescope were invented in the 1600s. Telescope magnifies distant objects, whereas a microscope magnifies near objects. And these are tiny objects. It was the work of a number of scientists and the advances in glass and lens making that led to the development of the microscope. Robert Hooke. In 1665, Robert Hooke observed a cork with his microscope and called the structure cells because the structures reminded him of a honeycomb. He was actually observing cell walls of dead plant cells. Followed by him, we have Anton van Leeuwenhoek, and in 1674, he observed blood cells, bacteria, and other organisms, and drew them in amazing detail. So the light microscope. This is one that we have in the high school and that you have used. They are good for studying bacteria and live specimens. Their magnification power is limited to 2,000 times. In order to increase magnification, the lens had to be made thicker, which causes the light to bend and the image to be fuzzy. In order to reduce this fuzziness, a drop of oil placed between the objective lens and the slide reduces the bending of light rays and thus improves the situation. The lenses are called oil immersions. They are not something that we do in high school, but if you go on to post-secondary, that is one of the techniques you will learn. Transmission electron microscope, also known as TEM. In 1938, at U of T, University of Toronto, James Hiller and Albus Prebus developed the first electron microscope, which had greater resolution. Resolution refers to the clarity of the image. So, therefore, with greater resolution, you have greater clarity of the image. TEM uses a beam of electrons instead of a light source, and it's capable of magnifying 2 million times. So this is much bigger than the 2,000 times that a light microscope can. The images are sharp, but the specimen is dead. They are embedded in plastic and can only be about 0.1 to 1 micron thick. And remember, a micron is a thousandth of a millimeter. Cell structures can be studied with a transmission electron microscope. This would be an example of a transmission electron microscope. It's not a microscope that we use at high school level. Scanning electron microscope, also known as SEM. It produces a 3D image as electrons scan the exterior surface of the specimen. This causes the specimen to release electrons, which are captured to form a three-dimensional image. Specimens may be coated in gold dust to produce a sharper image. Magnification power for a scanning electron microscope is not great. It's only about 200 to 500 times, so it's even less detailed than what you see in your light microscope. But the image itself is detailed. So this is an example of what it would look like, and it would come up 3D image on a computer screen. A scanning tunneling microscope, STM, is a minuscule electron probe that is placed near the surface of the sample, and a topographical image is created by a computer. The images are produced at an atomic level and are used to manipulate molecules and atoms. So they're looking at the atomic level. So you're looking at all the different atoms and the number of atoms in that specimen. And so it's a very large machine, and then it will come up on a screen like a topographic map. So what's the value in this? Well, microscopes allow scientists to study the structure of cells. Other technologies had to be employed to study their function. Cell fractionation is a process where cells are broken up and then the cell contents are spun in a centrifuge. The cell parts separate into layers depending upon their weight. The layers can be removed, and chemical reactions that occur in each layer can be studied. Radioisotopes. Living cells can be treated with radioisotopes. Isotopes are atoms of the same element that have different atomic masses because they contain different number of neutrons. So, for example, you sometimes have carbon-12 or carbon-14. They have the same amount of electrons and protons, but their neutron value is different. Some isotopes are radioactive, which means they can give off radiation that can be traced with special equipment. Uranium, 238, 237. The movement of the radioactive element as they precipitate in the chemical reactions in the cell helps science determine the function of each cell part or organelle.